Hello, everyone, and welcome to today's presentation titled Advanced Solutions for Gene Therapy Development. This webinar is part of the 7th annual event in the Bioprocessing Virtual Event Series. I am Melanie Chan of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by GenScript. For more information about GenScript, go to www.genscript.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat feature located at the left of your screen. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now, let's get started. I now present today's speaker, Roumen Bogoev, Head of Segment Marketing at GenScript. For a complete biography on our speakers, please visit the Presenter tab from the menu on the left of your screen. Roumen, you may now begin your presentation.
Thank you, Melanie, for the introduction, for the kind introduction. Today, I will focus on our advanced solutions for gene therapy development. Well, I'll be discussing some of the portfolio that we have for supporting development of gene therapies. I'll focus mostly on more advanced technologies that we are offering to our customers that are more focused on newer developments in how gene therapies are being developed and advanced. So this is the agenda for today. And first, a little bit about GenScript. So GenScript is a global company. We operate in over 100 countries around the world. We service customers in over 100 countries around the world. We have serviced over 200,000 customers with our services and products. We have over 6,000 employees. We have more than 800 people R&D staff. We have many patents and patent applications, and we have more than 100,000 citations using our products and services. We have multiple manufacturing sites. We have two in United States, one in the East Coast, New Jersey, and another one in Washington State in the West Coast. We have multiple manufacturing sites in China, and we have manufacturing site in Singapore. We also have many commercial offices around the world, several in Europe, some in Asia, Australia, and so on. So the GenScript group consists of several entities. I'll start from the right side. Bestzyme is our synthetic biology company. They have innovative capabilities in that area. They have very large capacity, and they focus on human and animal food supply. Legend Biotech, they're a cell therapy company. They became public a couple of years ago on the New York Stock Exchange. They develop innovative cell therapies, and they launched a couple of years ago, they launched Carvykti for multiple myeloma, working with Janssen. They have also advanced pipeline of therapeutics for developing for leukemia, solid tumors, infectious disease, and others. Today, my talk, however, will focus on the entities on the left side, which are our life science group. The life science group contains our services division and our product division. They offer reliable services and products for therapeutic development and for research. We use a lot of automation in our services operations to boost our efficiency. We also offer a lot of cutting-edge services and products, and some of these products are also focused on automating various processes in the research. ProBio is our biologic CDMO. They have very large GMP capacity. They have very wide offering in various areas of biologics developments. They have very strong regulatory experience, and they're one of the leading biologic CDMO service providers in the world. So first, a little bit about gene editing. That is one of the primary technologies used for gene therapies. So gene editing can be done in vivo or ex vivo. When it's done in vivo, usually the therapeutic agents are delivered using some kind of delivery methodology, could be viral-based or non-viral-based, lipid nanoparticle or others. It's delivered to the human body where the editing of the genes is performed. For ex vivo gene editing, usually cells are extracted from the patient, then they're modified ex vivo outside of the patient body using various methodologies, again, and then infused back into the patient's body. Today, my discussion will focus mostly on our portfolio for in vivo gene editing, because I have a colleague of mine, Daksha Patel, she is presenting on our technologies for cell therapies. So if somebody is interested in that, please look up her webinar also. So looking at gene editing technologies, some of the first targeted gene editing technologies developed was the zinc finger proteins. Those are proteins that can bind to specific DNA sequence, and when they bind to that sequence, they have also a nuclease attached to the protein, and the nuclease cleaves the DNA sequence, and then it's actually edited. Another developed technology is TALENs, using TALENs or tail domains. The tail domains bind also to specific DNA sequence, and they also have nuclease attached to them. Cleavage is performed, and then the editing is performed basically for this position. However, these two methodologies, TALENs, for example, they work well, they're easy to target. However, you have to develop new proteins, new domains for every sequence that you would like to edit. So that's very time-consuming and problematic. While with CRISPR-Cas9, which was developed a little bit later, you have universal enzyme, the Cas enzyme that is doing the editing, and the targeting is done with the targeting short oligo sequence that is used for targeting binding to the enzyme and binding to the DNA sequence. So that makes it much more convenient, and therefore CRISPR has become pretty much the premier methodologies for gene editing for gene therapies. So my discussion going forward will be focused on our CRISPR portfolio and some related products and services to it. So CRISPR gene editing can be done using different enzymes. There are a variety of enzymes being utilized and more discovered and added to the portfolios, to the methodology. Cas9 DNA editing was one of the first that was invented and then started being used in research. It's characterized with the fact that it performs double-strand bond break of the DNA and cleaves usually about 3 bases away from the PAM sequence. And then after cleavage, the enzyme is released and usually prefers G-rich PAM sequences. Cas12a is another enzyme that was also introduced for CRISPR gene editing. The characteristic for it is that actually makes double-stranded staggered break that allows for Ki insertion. You can actually have positioning, proper positioning of the fragment because of the staggered break. So that improves the Ki experiments. Cleaves about 18 to 23 bases downstream, stays bound after the cleavage, and usually prefers T-rich PAM sequences. Then Cas13a is RNA used for RNA editing. Is the single-stranded break, cleaves downstream, stays bound after the cleavage. So there are different enzymes that can be used for DNA or for RNA. There is also a wider range of enzymes. I'll mention some of them a little bit later. Some new modalities that have been developed recently by Broad Institute are the base editing and prime editing. They differ from the conventional CRISPR-Cas mediated editing by the fact that usually the break on the DNA is single-stranded break. So usually to perform this methodology, usually a Cas9 nickase is bound to another enzyme to perform the editing. So in case of the base editing, the Cas9 nickase is linked to deamidase, and when it cleaves the DNA strand, the deamidates the base and is converted to another one. So, for example, C and G bases can be converted to TNA, and T bases can be converted to G and C. For prime editing, is achieved a little bit differently. Cas9 nickase is bound to reverse transcriptase, And when it makes the break, there is also a longer gRNA being used called pecRNA that provides also some of the template for this additional sequence to be added or replaced during the editing. So in that case, basically a cleavage is done, then some new sequence could be synthesized using this template, part of the pecRNA, and DNA is edited. So if we look at the different pros and cons of the different methodologies, Cas9 is very popular methodology. It's one of the ones that researchers have the most experience with. It's also very flexible. You can do a large range of edits, deletions, insertions, modifications. It's pretty easy to use, to target. It really excels in large scale insertions because you can insert a full gene, for example. Concert that is not that specific. It still has the propensity to do some non-specific cleavages and non-specific edits. And there are a number of Cas enzymes now being developed to try to improve on that issue. Base editing is more specific and has fewer issues with unintended modifications because it does the single-strand cleavage. It's much more efficient than Cas9 at editing single bases. However, when you're editing only single base, only 50% of the conditions are due to single base mutations. So you can only develop therapeutics for 50% of the conditions, and other ones you cannot develop therapeutics using this methodology. Another issue sometimes is that neighboring Cs and As can also be converted, so that leads to some non-specific modification. Also, the enzyme is pretty large since you have the nickase linked to the amidase, becomes pretty large enzyme and creates difficulties with some of the delivery methodologies. Prime editing is, again, also more specific typically than Cas9. Is a little bit more flexible than base editing because it allows also for insertions of sequences. However, these insertions are typically limited to less than 50 bases that can be inserted. And it's also a relatively large enzyme that also requires a little bit more work to be transfected. So GenScript has very wide range of services and products that can service customers developing gene therapies. Anything from starting from target discovery and validation. We have a lot of services and products like gene synthesis, PCR agents, Western blotting, very strong Western blotting, electrophoresis portfolio, plasmid production, antibody development, CRISPR libraries, many tools that can be used for discovery and validation. We offer them. Lead generation and optimization, we have very strong portfolio for designing leads and optimizing them. sgRNAs, cRNAs, pecRNAs can be synthesized. Cas enzymes, variety of them. I'll show you a little bit later more detail on that. HDR templates, we have wide variety of those. CRISPR kits for optimization. We offer IVT mRNA, LNP production, AAV engineering. We have also some plasmid purification instruments I will mention a little bit later. So very wide range portfolio for lead generation and optimization. For preclinical work, we also have some preclinical grade services and products. Sometimes we call them GMP-like. sgRNA, cRNA, pecRNA, ISO quality, ISO 13485 quality Cas enzymes, GMP-like HDR templates, AAV packaging. For clinical work, we have a lot of cGMP services and products that can be used, RNAs, sgRNAs, Cas enzymes, HDR, AAV, and so on. So in this discussion, however, in this presentation, I'll focus only on the gene editing portfolio. So I'm not going to touch on some of the services and products that we have for Western blotting, antibody development, stuff like that. I'm not going to talk about those. And again, as I mentioned, I'll focus more on the newer services and products that we have supporting novel technologies for gene therapy development. So for gene editing specifically, which is one of the primary methodologies used for gene therapies, we have offering that can support anything from discovery through preclinical development and clinical trials, and then GMP production. We have various grades of services and products. As I mentioned, Cas enzymes, HgRNAs, HDR templates in various grades and various types, including AAV and LNPs for delivery. So we have pretty strong portfolio of off-the-shelf enzymes for CRISPR gene editing. We have several different Cas9 enzymes. We have the SP-Cas9, SP-Cas9 with different amount of NLSs and with TAC and without TAC. We have ESP-Cas9, which is very high specificity enzyme, also offered as REO and GMP grade. We have also enzyme SP-Cas9 with GFP fusion that can be used for tracking of your transfection. And we have also variety of Cas12a enzymes and some variety of Cas13a enzymes. So we have pretty wide range of enzymes that can be used for your CRISPR editing methodologies. I'll just highlight a little bit our ESP-Cas9 is very high specificity enzyme, very high fidelity. We compared it actually with other high fidelity offerings on the market, for example, for knockout efficiency in HEK293 T cells. And you can see that our enzyme here on the right side compares very favorably to the high fidelity enzymes from other vendors. And for knock-in efficiency also, we compared with high fidelity enzymes sold by another vendor, and you can see that our ESP-Cas9 compares very favorably, in some ways maybe slightly better than the high fidelity enzymes from other vendors. In addition to the regular Cas9, Cas12, and Cas13 enzymes, we also started adding some of the more novel enzymes to our portfolio. So with license from Broad Institute, we have started adding some of the prime editing enzymes and base editing enzymes that they're using for their advanced methodologies. So those are now off-the-shelf offerings that you can purchase and receive right away. We have, for example, P6C prime editor optimized for prime editing in plant genomes, P6D prime editor, enhanced editing accuracy minimizing byproducts, P-Max prime editor optimized by removing RNase activity for increased editing efficiency. We have also some of the original P2 prime editor, Cas9 nickase linked to reverse transcriptase, some of the original formulations brought, developed. And we have also a base editor that converts A and T to G to C. And these are offered as proteins, as enzymes. We also have some of these offerings in mRNA format, which I will mention a little bit later. In addition to off-the-shelf enzymes, we also, of course, very strong at developing custom. So we have very strong capabilities to develop enzymes or optimize them. We have very strong machine learning and AI capabilities to support that development and optimization. And we can develop a lot of different, we have developed already very wide range of CRISPR enzymes, including some of the base editors, prime editors, and so on, some of the more novel enzymes. So for knock-in HDR templates, we offer a variety of those with different properties. So gene-exact single-stranded DNA is one of our offerings for HDR template. Is very useful usually for T-cell therapies, also for cell and animal model generation. The nice thing for this template is that it has minimal cytotoxicity, allows for precise knock-in, minimized off-target effects. The limitation for this template is that it's actually the length of the template can be only up to 5 kilobases, so 115 nucleotides to 5 kilobases. So it's limited a little bit in the size. Otherwise, we have high variety of purities and sequences and microgram to milligram scale quantity. We have also research to CGMP grade. Another template that we offer is the double-stranded gene-1 double-stranded DNAs with covalently closed ends. Basically, this allows for longer insertion of longer fragments. It's also very suitable for scale-up. So the ends are covalently close to make it more stable. It's more suitable for scale up. Insertion length can be up to 10 kilobases for this HDR template. And we offer it in high purity sequence verified, again, microgram to gram scale, and we have research and GMP grade that we can offer. GenCircle double-stranded DNA is one of our newer and more advanced templates. It's basically based on a very small vector that is 429 base pairs vector backbone. It doesn't have any antibiotic resistance, have very low cytotoxicity, usually leads to better KI performance, usually typical increase about 30% better. It's ideal for very large insertions and replacing regular plasmids as a HDR template. Insertion length can be up to 50 kilobases. And again, we offer different quantities, and we can offer research or GMP grade of this template. Here is just example of using GenCircle double-stranded DNA instead of a plasmid. So let's say when we compare the GenCircle compared to a plasmid, you can see here on the chart below, you can see that it provides much better expression compared to regular plasmid, much better knock-in efficiency compared to regular plasmid. Here is another example of GenCircle being used as transposon vector for knock-in. And again, comparing with PAC vector, you can see that GenCircle provides much better knock-in efficiency, even at different quantities of the vector being used, consistently better, significantly better performance than PAC vector. We, of course, can synthesize variety of different gRNAs for the different enzymes. We have several different grades. Easy Edit, which is our most economical offerings. It offers high editing efficiency, flexibility in scale. Safe Edit is our higher grade, which is HPLC purified. It minimizes impact on cell viability and provides reduced off-target editing. And of course, we can offer gRNA synthesis, GMP-like and cGMP grade for IND filings to clinical trials and clinical manufacturing. In addition to that, of course, we can synthesize pegRNAs for prime editing. We can synthesize them with up to 266 nucleotides length. I believe we can offer the longest length from any vendor on the market. And up to 200 different modifications can be done in order to meet your requirements. In addition to synthesizing custom sgRNA, we have very strong database of pre-validated sgRNAs we are licensed from Broad Institute, basically either for knockout or transcriptional activation with basically every human gene available. We have multiple sgRNAs that can be utilized for either knockout or transcriptional activation. We also offer CRISPR cell line services. We can do knockouts, single gene or multiple genes. We can do knock-ins, variety of those for different purpose. And we can do full whole genome deletion service also. And they have on the very right side here are the different timelines, depends on the complexity of the service and the cell line selected. So lately, mRNA is becoming more and more popular as delivery methodology, delivery format for gene editing, mostly due to its transient nature, because it allows for the gene editing to be performed and then the gene editing reagents to be degraded pretty quickly, instead of lingering in the human body and maybe causing some non-specific gene edits because of the very long residual time. So we have full offering from beginning to end. We can start with codon optimization. We have very strong and very easy to use software for codon optimization, considering over 200 different factors. We have also pre-designed UTR regions that we can recommend, or we can use the customer preferred UTR regions. We can do the gene synthesis, of course, anything from 100 base pairs to 20 kilobases genes with relatively short turnaround time. We have special vectors with built-in poly-A tails to support the poly-A integrity and precise length. We also can do the cloning and plasmid preparation, including plasmid linearization. We have vectors that already encoded poly-A tails. We can provide development of custom enzymes. We also develop a lot of our own proprietary enzymes for polymerases and capping enzymes. We can provide variety of mRNA of RNA formats, anything from linear RNA, circular RNA, saRNA. I'll expand a little bit more on our service offerings for those a little bit later. But we can offer any of these formats, and we can offer them with various modifications and various purity formats. In addition, we can also prepare lipid nanoparticles in various quantities. And I'll expand a little bit more on this service also going forward. So for linear mRNA, we have also poly-A tail solutions that allow us to design the poly-A tail and keep it at certain specific length based on the customer requirements and with high integrity. One of the new services that we started offering last year, and we're expanding a little bit further on those, are around the circular RNA. So circular RNA is a novel class of RNA molecules that is being used now for therapeutics, vaccines. Some of them have coding functions. So if they have coding function, they're usually good for therapeutics and vaccines, and they provide better stability for longer expression duration. They typically offer higher protein expression, and LNPs using circular RNA is more thermostable, so they can last longer. They could be also used as non-coding RNAs for therapeutics, as miRNA, protein sponges, siRNA, and so on. So they can have coding or non-coding function. And we're one of the first vendors on the market that offers circular RNA, and now we're going to expand even further on this offering, making it even stronger. Circular RNA has, as I mentioned, advantages that provides typically stronger expression, requires lower dosage for therapeutics. It's more stable. If you look at the copy number of circular RNA versus linear RNA, you're going to see higher copy number usually after 24 hours or 72 hours compared to the linear mRNA. And if we look more specifically at the differences between circular RNA and linear mRNA, we're going to see that basically circular RNA doesn't have open ends. They are circularized, so there's no 5' and 3' open ends. So that prevents some of the exonuclease degradation. It includes also ribosomal binding site. So that's a must because it doesn't have the 5' cap anymore for ribosomal binding. And basically includes the ribosomal binding site, exon, linker sequence, and then the sequence of interest. We can offer it now. Our new offering is until now, we could offer only the scarred version of it with some extra sequence in there. And now we are able to offer also scarless circular RNA, and I'll show you more about that in the next slide. And the linear, of course, has the two open ends. We also have multiple, usually those are capped for increasing the stability. We have multiple capping options for that. We can also do codon optimization in different modifications. And we can provide basically recommended UTR regions and stuff like that. But typically, the circular RNA provides a little bit better performance in most cases. So, for example, if we look at our circular RNA, until now, we have been offering the one on the left, basically, circular RNA with SCAR using Pi technology. Basically, those are usually used for protein translation in coding applications and generate circular RNA with small SCAR sequence. They can be up to 5 kilobases long and includes the iris region, exon, linker, SCAR sequence, and ORF gene. Now our new offering offers you this without the SCAR sequence. So it's only basically the iris region and and the gene of interest. So in that case, you do not have any of these extra elements. It still can be up to 5 kilobases long, and this makes it much more stable. These two formats, the left one and the one in the middle, they are used usually for expression applications. So they are encoding a protein for expression. We can offer also scarless circular RNA using T4 ligation, where we can enclose essentially the sequence that the customer provides us without any extra elements. This one does not include the iris region, so it's not for expression applications. This one is only for non-coding applications. This one can be up to 2 kilobases long, and we basically can circularize the customer provided sequence. So if we look at the performance of different types of formats of mRNA, we can see that linear mRNA degrades relatively quickly. Usually the expression is moderate expression with a relatively quick decrease in the expression with the time. Circular RNA is much more stable. Even with a scarce sequence in there, you can see that the expression level continues until day 6, increasing until day 6, and then starts declining in day 8. If you look at circular RNA scarless, actually it's even more stable than that. It provides even higher expression, and also it continues increasing the expression level until day 8. So it's much more stable than the scarred sequence. So this is in HEK293 T cells. If we look at Jurkat cells, the picture looks a little bit different, but still the scarless format offers stronger expression in the long term, with preserving the expression levels much better than the scarred sequence. So in addition to services, we started generating some off-the-shelf products. We think that could be very convenient for our customers, sometimes to use them as positive controls, sometimes for various experiments. And the nice thing is this offer you very fast delivery. Those are ready to use, already QC'd. And basically we offer several of those: EGFP, FLUC, EGFP scarless circular RNA ladder. So we're the only vendor on the market at the moment that offers circular RNA ladders. So if you need any of those, look them up on our website. So if we compare, we also offer self-amplifying RNA, which is another format that could be used for various applications. The self-amplifying RNA, of course, include the replication gene, so it allows it to create more copies while it's being expressed in the cell, and that gives you much stronger expression. So if we compare the performance of different types of different formats of mRNA, we can see the linear RNA has certain expression, circular RNA has stronger expression, and then the self-amplifying RNA, even at half of the amount being used for the others, give you much stronger expression at 48 and even 72 hours. So you can see the benefits of using self-amplifying RNA also. As I mentioned, we started introducing more and more off-the-shelf products for customer convenience that gives you very fast delivery. And we have some products that use linear mRNA, some use circular RNA, some use self-amplifying RNA. We also have some LNP formulations that are off the shelf, regular LNPs, targeted LNPs. They follow the off-the-shelf product follow in 3 categories. They're either ladders that you can use for your measurements, or they're reporter genes, something like EGFP, like mCherry, like FLUC, or they're gene editing, basically gene editing Cas enzymes or prime editing enzymes. I showed only a few of those as illustration. We have some of the base editors, some of the prime editors also in mRNA format. We also have a lot of the Cas9s, Cas12s, and other Cas enzymes as in mRNA format. So those can be used for your experiments. So this is a little bit of the customer feedback that we have received so far. Customers have used our mRNA. Basically on the left side, the customer used it for for EGFP expression in cells. with good success. Some of our mRNA has been used by customers also for in vivo experiments. You can see in mice, results in mice here in the middle section. And we have some, basically, some of our customers like Merck and XAP have expressed their satisfaction with using our mRNA for different applications. So mRNA can be delivered in different ways into the cell. There was a paper analyzing over 260 different mRNA-based therapeutics with disclosed delivery systems, and what they found out was that actually majority of those are delivered using some kind of nanoparticle. Usually lipid nanoparticles is the most prominent one. Some of them are delivered with liposomal delivery, exosomal delivery, adeno-associated virus, and then small percentage with peptides. But by far the biggest one is with lipid nanoparticles. Because of this prominence of the lipid nanoparticles for delivery, we have also developed lipid nanoparticle service. One of those service options is our radiated LNP service. Basically, it starts from selecting basically the editing system that you would like to use. Is it Cas9, Cas12, base or prime editors that we can synthesize in the desired format. Build basically your elements that you need, any kind of sgRNAs, DNA templates and stuff like that. And then making the LNP formulations based on your specifications, and then QCing and validating those LNPs. So this is all included in the radiated LNP service. In addition, this is some examples just showing radiated LNP service in knockout using different formulations, and also knock-in service with TGFP. You can see the higher expression here after the knock-in. In addition to regular LNP services, we also offer targeted options. Some of the targeted options are pre-optimized. We have already pre-selected certain formulations that work very well for certain type of cells or for certain application. For example, we have CD3 cell-targeted LNP, CD34 cell-targeted LNP using antibodies. We have liver-targeted LNPs, we have spleen-targeted LNPs, lung-targeted LNPs. So those are pre-designed formulations that you can just order. In addition, of course, we can work with you to develop any of your desired targeted LNP formulations and further optimize them. We have very extensive QC. We have different size options for different inserts and different amounts of LNPs and mRNAs. And we can also prepare them for different type of administration, intramuscular, intravenous, for in vitro studies, and so on. In addition to LNPs, of course, viral vectors are very often used. A lot of the existing gene therapies actually are using viral vectors for the delivery. They're very efficient as a delivery system. Some of the challenges with viral vectors, however, especially when it comes down to newer technologies that are being used right now, like the prime editing and base editing, is that actually the size of these new editors exceeds the genome size of the AAV. So in such case, maybe another virus can be used, either lenti or adenovirus or another virus that can have bigger size genome. But this is a challenge because AAV is one of the more prominent delivery vehicles for gene therapies by far compared to the others. So because of the need for AAV viruses for delivery, we also offer AAV engineering service, and we can start, of course, with the codon optimization. As I mentioned before, we have very good codon optimization tool, looking at over 200 different variances that can be considered. The gene can be either synthesized or you can purchase it from our ORF cDNA clone collection. Then we can engineer the vector accordingly. We can add different regulatory elements in the vector. We also guarantee our AAV ITR stability. We guarantee that. We can also engineer the capsids for tropism, for delivery in specific tissues, using basically mutagenesis, or we can offer you mutagenesis libraries if you prefer to do it yourself. As I mentioned, we have different entities in the company that can service customers through different times of the development process. GenScript usually our LSG, our life science division services customers up to the preclinical stage. And we have some also GMP services under GenScript also. But a lot of the GMP services for gene therapy fall with our ProBio CDMO division. And they have very strong offerings here for plasmid CDMO services, all the plasmids needed for either AAV or lenti or other viruses. They offer also viral packages, lentivirus, AAV virus, CDMO service packages for that. They have very strong regulatory experience and GMP experience. They have done over 100 batches for different GMP products. They have passed 22 audits from various countries around the world in China, US, Europe. They have supported over 30 IND projects and have successfully supported customers to successfully submit 6 IND projects. And this is just a chart showing some of the other offerings that I didn't mention, a kind of summary of what we offer. These are still not all the services and products we offer, but some of the key ones that we offer. So antibody and protein expression, we're very strong in that area. We have a number of different expression systems, starting from E. coli, HEC293, baculovirus. But we have specifically optimized our CHO system, and we call it TurboCHO. We have optimized to be very efficient in expression and very fast. Some of the expressions can be done within 5 business days, starting from gene sequence all the way through purified protein. And we have it in research grade, and we have it in preclinical and also clinical GMP grade. CRISPR, gRNAs, and HDR templates, research grade, preclinical and clinical grade, plasmid preparation, research grade, preclinical and clinical grade, IVT mRNA, we have research grade and preclinical grade, GMP grade is coming soon, AAV, viral vector service, RUO, preclinical and clinical grades, lentivirus, similar thing, RUO, preclinical and clinical grade, peptide synthesis, research grade, and GMP grade. We have also, in addition to the services, we have some very innovative products. So we have cell isolation system, which includes automated instrumentation and magnetic beads and other consumables needed for the cell isolation, for cell therapies. As I mentioned, my colleague presenting on the cell therapies will give you more information on that. But basically, we offer it as a research grade, also as GMP grade. We have excellent T-cell activation reagent, also offered as a research grade and GMP grade. We have also some very novel plasmid purification systems. I'll show you a little bit more detail on that in the next few slides. They're offered mostly for research grade, automating large-scale plasmid purifications. And we have very innovative protein purification system, very easy to use and high throughput. And just a little bit about our new plasmid prep purifications. As I mentioned before, we also offer, of course, the service for plasmid prep, anything from growing the cells to isolation, purification, and QC, and delivery to you. However, if you're doing any plasmid purification in-house manually, then you may want to consider automating them. So this AMMA 4 Mini is automating the mini preps for plasmid prep. This is a relatively new addition to our portfolio. It was launched a few months ago. It's a modular system, so every module can purify 48 samples. 4 modules can be connected to one controller, so that can give you up to 192 sample preps for every 2 hours. It's fully automated. Again, it's modular design. You can scale according to your needs from 48 to 192 preps. Offer transfection quality plasmids. It's easy to set up, easy to operate, uses prepackaged consumables. So it's very simple and easy and effective system to automate your process. We also have a larger scale system, which is for MaxiPrep. The MaxiPrep system, we have been selling it for more than a couple of years now, and many companies have already purchased this system to automate their process. It's also fully automated. It's also modular. Every module can purify up to 6 MaxiPrep at a time. 4 modules can be connected to one controller, giving you up to 24 samples at a time. So 24 MaxiPreps in less than 2 hours. Provides transfection quality plasmids, uses prepackaged consumable cartridges, and is easy to set up and very easy to operate. So these are some of our newer product offerings that are very much related to gene therapy, since plasmids are so frequently used in many methodologies. So why using GenScript? GenScript is the number one service provider in the world for gene synthesis. We have been around since 2002. We have very strong expertise in anything from gene synthesis, gene editing, mutagenesis and cloning, gene variant libraries, and many other services and products that are needed for research and therapeutic development. We have serviced hundreds of thousands of customers in over 100 countries. We have synthesized over 3 million genes. We have more than 95% success rate. So we're very experienced in genomic research. And we pride ourselves with supporting our customers in their research. Our customers have published many papers citing our services and products. We have over 100,000 citations in very prominent journals like Cell, Nature, PNAS, and so on. And we recently expanded our GMP site. We recently developed a GMP site in Hopewell, New Jersey, in the US. That's our newest addition to our GMP facilities. Right now, this facility offers plasmid preparation, plasmid development and preparation in GMP grade. Very soon, we'll be offering also AAV and antivirus services in GMP. And we have also other GMP sites, with our largest GMP site being in Zhenjiang, China, 400,000 square feet with many different manufacturing lines and many different capabilities. And thank you for your attention. And now I'll be happy to answer any questions you may have.
Thank you, Roumen, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just submit a question in the Ask a Question box located at the bottom of your screen. Looks like we had quite a few questions come in already, so let's get started. Our first question asks, is the delivery timeline different for scarless versus scarred circRNA?
They're very similar. They're about 3 weeks for delivery, either the scarred version or the scarless version. Of course, some of the delivery timelines depends on the complexity of the project, how big are the inserts, and what is the scale of the production. But 3 weeks is kind of the typical for most of the projects.
Okay, thank you for that, Roumen. Our next question actually asks, can you make antibody-targeted LNPs?
Yes, yeah. So we have already developed 2 LNPs that we have pre-made formulations, CD3 antibody-targeted LNPs and CD34 antibody-targeted LNPs. And we would be happy to work with any customer to develop. If we have to, we can also develop the antibodies and develop the LNPs for that.
That's great. The next question we received asks, can you produce custom CRISPR-Cas enzymes?
Yes, so we develop all the time a lot of new enzymes for our own use and for our off-the-shelf offerings. We also do a lot of custom work. We have very strong machine learning and AI capabilities that help us support these developments and make us more efficient. So yes, we can develop new and we can produce them in various grade and research grade and GMP grade.
Okay, great. And so it looks like we have time for one final question here, and it asks, how can I order the circRNA? Excuse me.
So the circular RNA now can be ordered online. We have very easy to use form that lets you select all the needed traits and modifications that you like. We are the only vendor at the moment that offers this as convenience to our customers. Of course, you are always welcome to call our customer service. However, that can be done easily online.
Okay, thank you, Roumen. Do you have any final comments for our audience members here today?
So I would just like to thank everybody for their attention, and please feel free to send me your questions by email if you have any further questions, and feel free to go to our website and learn more about our new offerings. And thank you again.
Awesome. Yeah, thank you, Roumen Bogoev, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for your interesting questions. I'd also like to thank our sponsor, GenScript, for sponsoring today's webinar. Please visit GenScript's booth in the exhibit hall after the webinar for more information and resources. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until April 2nd, 2026. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's lab event. Until next time, goodbye.